So we've talked about all of the conditions that have to be met for a population to not be evolving. So keep in mind that most populations are evolving at any given point in time, and that's because one or more of those conditions are not met. But that doesn't mean that Hardy-Weinberg equilibrium isn't useful. There is actually an equation that goes along with Hardy-Weinberg equilibrium that allows us to calculate allele frequencies at a specific point in time. And so we're sort of assuming that at a single instant in time, no evolution is happening. And then at another single instant in time, no evolution is happening. And so you can use that information to compare to points in time to see if allele frequencies have changed. Now, I guarantee that I will ask you to use this equation on the test. And I guarantee that this is the most missed problem on the test because students do not take the time to pay attention and learn the difference between allele frequencies and genotype frequencies. And this problem tends to be a hit or miss problem. If you understand allele frequencies, you get it. If you don't understand where you get allele frequencies, you're going to have a problem. So you are going to have to take the time to take notes on this problem, to take notes on the practice problem, to ask questions in class, to pay attention to what's going on. The math is not hard at all. But students get lost in the two little details, the difference between what an allele frequency is and what a genotype frequency is. So let's use this Hardy-Weinberg equation to calculate some allele frequencies with the idea that we could compare them to a later generation to see if those allele frequencies have changed over time. Because remember, our present day definition of evolution and of microevolution is that evolution is a change in allele frequencies over time. So here is our glorious Hardy-Weinberg equation. P squared plus 2pq plus q squared equals 1. There's supposed to be an equals 1 there. And magically there it is. P squared plus 2pq plus q squared equals 1. So I'm sure you are thinking, what in the heck does all that mean? Well, it's a really simple equation. It's actually a binomial expansion of a plus b equals 1. If you don't know what that is, you don't know how to do it, that's quite all right. I'm not going to ask you to do it. I'm just going to ask you to remember that this equation is p squared plus 2p squared plus 2pq plus q squared equals 1. So let's look at 500 wildflowers. So these are those same pink, red, and white wildflowers that we saw in the genetic drift example. And then we're going to calculate, we're going to look at the allele frequencies. So P and Q are going to be equal to the frequency of the allele for two different alleles in a population. So in this situation, we have a red allele and we have a white allele. Now this could just easily be a dominant recessive allele where P is equal to the frequency of the dominant allele, Q is equal to the frequency of the recessive allele. In our situation, we actually have incomplete dominance, which is where you get red and white coming together to make a pink individual. So in this case, you can actually see three different genotypes. You can see the homozygous dominant, the heterozygous, and the recessive homozygous recessive phenotype, and you can use that information to calculate the allele frequencies in a population. This is where students get confused. So I just talked about the allele frequency. The allele frequency is the number of a particular allele divided by the total number of alleles in a population. This is how you calculate your grade on a test. You calculate how many points you have out of the total. You generally multiply by 100 to get a percentage, but we're going to keep it as a decimal point. Allele frequencies are based on the number of alleles that are present in a population. And you can find that by looking at the phenotypes of an individual. Now, students get confused because the equation itself is actually asking you to know the genotypes of the individuals, not the allele frequencies. You do have to know the allele frequencies, but the equation is actually telling you expected genotype frequencies. So our p squared is equal to one of the homozygous genotypes, p squared. 2pq is the expected heterozygous frequency, so genotype frequency of the heterozygote, and q squared is the expected genotype frequency for the other allele. Now this is all equal to 1. Everything is going to have to add up to 1, and that's important. We can use that information in just a little bit. So you can derive this formula mathematically, or you can think about the genetics behind it. So remember, if we have a homozygous dominant allele, it is the chance of getting that homozygous dominant allele is the chance of getting that allele from mom and the chance of getting the allele from dad. So a couple of slides ago, I said that the frequency of the P allele, which is representing the red allele here, is 0.8. So the chance of getting two homozygous alleles in a population is 0.8 times 0.8. So that is your P squared, 0.8 times 0.8, or the chance of getting one P allele and two P alleles. So now the same thing is true for the Q allele. The chance of getting two of the same alleles in the population. So if the allele frequency of the white allele is 0.2, represented by Q, the chance of getting two of them is 0.2 times 0.2, or Q squared. Now the chance of getting a heterozygous situation actually can happen two ways. So that's why we multiply it by 2. It's the chance of getting a P allele and a Q allele. So we see that here, P allele, Q allele. So 0.8 times 0.2, chance of getting heterozygous is 0.6. We can also get this the other way. You can get a Q from the opposite parent and a P from the opposite parent. So the probability of that is P times Q. So two ways that you can get this. So P squared is the genotype frequency of the homozygous dominant. 2pq is the genotype frequency of the heterozygote, and q squared is the genotype frequency of the homozygous recessive. Now that is based on the frequency of each red and white allele, or the P and Q alleles in the population. Remember, having the allele affects having the genotype, and that is the difference in this situation and where students tend to get confused. So let's calculate some allele quantities. Let's discover how they actually calculated out of those 500 wildflowers that I showed in the beginning that P was equal to 0.8 and Q was equal to 0.2. I would strongly suggest that you go back and write down the equation, pause the video, label some things, label P, label Q, label your genotypes, and then come back and we'll go through this allele quantity calculation. So I'm glad you did that. I'm glad you took a break and wrote some things down, because now you're going to write down some more math. This is really just going to be me babbling some numbers, but if you get confused, I want you to pause and think about what I'm talking about. This is the exact example in your book, so if you're still confused, go back to your book, read their explanation. So we have 500 wildflowers, pink, red, and white. Remember that each individual has 2 alleles for every trait. These 2 alleles in this situation can either be the red allele or the white allele. That means that we have 1,000 alleles a total. So you have 500 wildflowers times 2 alleles each equals 1,000 alleles in the population. So let's say we have 320 red flowers, 160 pink flowers, pink apparently, and 20 white flowers. Let's calculate the frequencies of the red allele and the white allele. You do that by dividing the number of alleles for whichever color you're interested in, divided by the total number of alleles in your population. So we have to think about where those alleles come from. If we have a red flower, each individual has 2 copies of the red allele. So if we have 320 red alleles, that means we have 320 copies of one allele 320 copies of the other allele. But we also have that red allele in our pink flower. Remember, they are our heterozygotes. So here, if we have 160 pink flowers, that means we have 160 red alleles because each individual has one red allele. So if we total that all up, we get 800 red alleles divided by 1,000 alleles total. So our p-value, the allele frequency of our red our red allele is 0.8. If you need to work through that on your own, pause now and take some time to do that. All right, unpause. So let's calculate the frequency of the white allele. This is going to be represented by the letter Q. So if we are a homozygous white individual, each individual has 2 copies of that white allele. So if there are 20 white individuals, we have 20 copies of one allele, 20 copies of the other allele. Again, remember that the individuals who are pink also have a copy of the white allele. So if we have 160 pink individuals, we have 160 white alleles for those pink individuals. Total those up, 200 white alleles total. Frequency of the white allele is 200 divided by total number of alleles, which is 1,000, giving the Q value of 0.2. Now, there are 2 ways actually to calculate Q or P. If you start with one, you can calculate the other. And that's because this is a frequency. Frequencies always must add up to 1. So P plus Q equals 1 for all alleles. The frequency of one allele plus the frequency of the other equals 1. So you can use that idea to calculate Q directly without actually calculating this allele frequency here. So if we do P plus Q equals 1, 1 minus P equals Q, so 1 minus 0.8 equals 0.2. So we can get Q that way as well. Please make sure that you understand this calculation both ways and understand why you can use P plus Q equals 1 instead of calculating the allele frequency directly.